at 11:10 until 11:50 before we would come back to the module 5B on the molecular dynamic. So please be mind about that. It's Bangkok time, so you might want to check what is it in your home timing. And then the quiz for all modules today will be given at the end of the day. Okay, so please note it to fill the quiz within the time given. Yes. So that's a change for today. So I hope you would be able to follow those, and maybe make sure that your lunch is there at 11:10. It's quite a short time, 40 minutes, but I hope you can make it. And you can always bring your lunch and your coffee in front of your computer, and then following the lectures. Okay. Um, yeah, it's 6 minutes to 9 a.m. Another thing, maybe I would like also to again remind some people here that haven't adjusted their names to initial country initials and then your full name. Please do that so that your lecturer can see where you are from and who you are, what your name is. Um, yeah, I think that's all the changes for today. Any questions from the students from the class about the adjustment? No, I guess all good. Okay, thank you. Oh, okay. One more thing. The presentation materials from Monday's sessions can already be downloaded on the hpcschool.net. So please do download. Um, yeah, I think that's it. If you don't have any questions, let me check the.
Yeah, I have a question. Good morning to everybody. Good morning, Intan.
Oh, good morning, Paldo. Yes.
I have a question to the students. If any of them experience some difficulties with their connection in these first 3 days of the class of the school, from Monday, or if everything went smooth for them.
Yeah. Some of I also got some chats that some of the students in the Philippines, because they had thunderstorm yesterday and maybe today as well. Yeah, so it might what is it? The connection might be a little bit rusty, and maybe other students here is having connection problems that want to be shared with us, or if you have any specific condition. All good. Okay. Okay, I think maybe all good still this morning, Paldo.
Good. No news, good news.
Yeah, I think so. No news, good news. Okay. Well, if there's anything that you would like to share with us about your condition, just feel free to use the chat box, as I said before. Okay. So I think we have our lecture here. Let me check.
Yes, I'm here. Hi, everyone.
Hi, Dr. Sebastian Morstraw. Hi. Let me ask the admin to spotlight you. Yes. Hi, Dr. Sebastian. How are you?
I'm good. How was the first day for everyone? I heard thunderstorms. I'm located in Singapore, so we also do get thunderstorms around.
Oh, okay, okay. So yeah, maybe is there any colleagues, any students from the class would like to chit chat a little bit? Maybe still having the coffee. So, have you got your breakfast already, Dr. Moristro?
Yes, and I also have a coffee.
Good, good, good. Great. Yeah, so I think we're 2 minutes
I think
before
that
nine.
there is a message.
Okay. Yes.
There is a message in the chat.
Mhm. Okay, where we can follow up the recording. I think at the end of the school, we're going to inform you about all the materials. So there will be an email blasted to all of the students about all the materials you can access. Um, and check the curriculum site, download slide link for the Lumi tutorial is not working. Okay, we will check on that. Thank you, Madam Joy. And then, okay, our team already said we will fix them. Okay, and good morning, everyone, said Miss Warawan from Thailand. Good morning, too. Okay, I think the class is ready now, Dr. Moristro. So we shall uh, start by now. Okay. So thank you, everyone. Good morning again. Before the class begin, I would like to introduce you to your lecture, your first lecture of the day, Dr. Sebastian Morris-Roth. He studied theoretical biochemistry at the University of Vienna and wrote his master and PhD thesis at the Institute of Molecular Pathology. After FEPS and Marie Curie Fellowship at the VIB Switch Lab in Brussels, he led a group of experts in protein sequence analysis as a senior principal investigator in the A-Star Bioinformatics Institute since 2007. He is the new executive director of BII from January 2021. His protein function analysis skills support A-star efforts at the public-private interface. Through computational analysis and modeling, his team is critically contributing to national and global viral pathogen surveillance. So without further ado, Dr. Morris Roy, the floor is yours. Thank you.
Thank you very much. Thanks for the introduction, and thank you also for the invitation, and thank you to all the students for your interest. I hope I can show you some interesting applications of high performance computing. Sometimes you can do several of the steps just on something as simple as your mobile phone, but what matters is how you use it for. So if you have a supercluster, if you're sending things to Fugaku, or if you can do a calculation on your mobile phone, all of it can be important. And I will show you examples across the spectrum. And as was I think mentioned before, the links to the slides that I will show to you, they will also be posted online already, I think, during the class or at least during your lunch break. And there will be a mini quiz, just a few questions, multiple choice. I think you may have had already some the other days. And you can use the slides to help answering that questions. I will also, during the presentation and tutorial, we will have 3 exercises that will give you a little hints what may be related to questions that come later. So it does pay off to pay attention. And yes, I'm very happy to share with you some of the work we are doing. And this pandemic that has affected all of us, including the ASEAN region, really changed a lot. It also changed a lot in the perception of people of the importance of bioinformatics and computational biology, the work you're interested in and the work we are all doing. And I want to start off with a little explanation. What do bioinformaticians do? And of course, several of you may already know this. I just want to give you a different perspective in how I see this. I use the same slide sometimes when I talk to biologists, people with biological background, to tell them how computers are useful for them. And in case, like here, where we have more students with background of computer science, I want to tell you the opposite. So your algorithms, your understanding, and the programs that you write, they can be important for biological problems. So what is important is that you understand a bit about the biological problem, and not necessarily understanding everything about the biology of it, but what is the problem that needs to be solved. And so in this slide here, we talk about how we understand biological, chemical, and medical data. And let me just put up here the laser pointer. And for example, you have a task. You need to sort these 5,000 papers into the correct drawer. And this drawer could be, for example, by a date. And if you do this manually, this takes very long. And the way you would sort things also computationally is you follow an algorithm. For example, for each paper in the pile and for each drawer in the database, you check if the dates or the time window matches. So if paper P belongs into the drawer, then you add it into the drawer. And you go through this, and later, for example, you're interested in which one was the most filled drawer. You sort the drawers by the highest paper count. So these are all algorithms that you commonly use. And what we need to do is to solve this task so the biological problem is formulated in a way the computer can understand it. And the algorithms are written as the computer programs, as you do. And there are many similarities in the way a bioinformatician works. For example, instead of the papers, you would have a list of thousands of genes, and the drawers are the pathways that they work in. So pathways are the connection of genes that do one function together, like the immune response, for example. And the very same analogy, like you would do it here, you can do it at the level of biological problems, like genes and pathways. And you can go both ways. If you're a computer scientist, you need to understand a bit, okay, the pathways, the immune response is one of the drawers. I want to see which gene belongs to that. Or if you're a biologist, I have this list of genes. How do I find out how they work together? And then you go to the algorithm side. And this workshop, also a lot of colleagues following after me, they will very nicely tell you about high performance applications in 3D protein structure. And my work is exactly at this interface, going from the sequence to the 3D protein structure. And again, here, what we do is, if you want to characterize a protein, its stability, we know that hydrophobic residues in amino acids behave like oil in water. They don't want to touch the water. They want to be inside. The driving force for a protein here to fold is to keep the oily hydrophobic residues on the inside and the watery hydrophilic residues on the outside, so the green ones. And if you know this biological concept, you can create an algorithm again to estimate the stability. And stability, for example, here can be calculated by checking how many of the gray ones are on the inside, how many of the green ones are on the outside. And if there is a mismatch, like a gray one on the outside, it becomes negative. And if there is a green one on the inside, it's also not good, then it's also negative. So my question to you, just a quick quiz question. Can you type in the chat, please, what are your estimates for the stability of this? What is S in this case? I want to do it a little bit interactive so that once in a while I will ask you some things, and the one who responds in the chat will get some extra points. So my question is, what is the score S here in this case? You can use this formula, and here is an example. So we have already two colleagues, Joshua and Varova and Ruchuta. So now a lot of people are answering. Very good. It is 6. Well calculated, including the formula. Very good. I like that many of you have got it together, and I will give to the first 3 extra points. The first 3 that write the number and the first 3 that write the whole formula, because you need longer to type. You'll get some extra points. I don't know exactly how they will calculate it together, and don't worry too much also about the exam question, because it's just a quiz. but I like very much to see your interaction here. So this is the way how we can use formulas and algorithms to solve biological problems. And this is what we most often do in bioinformatics. We are dealing with input data, sometimes in text, sometimes in binary. We write little programs, like this is just a Perl script. And we work on a command line using these formulas, describing how the protein is stable, how it folds, using the concept of what makes a protein fold. We combine this and we can make from visualization to stability calculation and a lot of other things. So this is day in and the day out work. And what I want to highlight to you now is that during this pandemic, I mentioned already, we've seen looking at sequences can be very important. So sequence analysis is very important. And in this case, this happens through national surveillance and global surveillance from WHO, as well as all the countries in ASEAN, all of your countries that you're from are contributing heavily to this. And the nice thing is we're all working together, sharing the sequences on a platform called GISAID. And why is it important to have this genome sequence and the genomic surveillance of the virus? First of all, you were able to see how a diagnostic kit works so that you can make a tool that can predict, or not predict, that can detect the virus. So that's the first important use. And then the way the virus is detected is with little probes. So sometimes there are mutations and you need to know if your probe can still detect it or if the mutation has happened there. Then the next question you can answer with the sequences is how is it spreading? You can compare the genomes with each other. So these are the things that I will mention here, sequence alignment and sequence similarity searches. And with that, you know what is where did the virus come from? What is the most closely related relative? And the early history of the outbreak, of course, you're interested from which kind of animal it came from. And but now we can go so far that you can even use it for contact tracing. Did the case in the shopping mall correspond to the case at the airport? These kind of questions can be answered. And very importantly, one of our main countermeasures, the vaccines, vaccine development started by the genomes being shared through the GISAID platform. Once you know the genome, these new technologies, including the mRNA vaccines, they could immediately take the information and start development of the new vaccine. And what we need to monitor now is we have variants occurring. And the question is, do the variants still work? And it's all very similar biological concepts that to understand, put into algorithms that allow us to do that. And this is the portal, GISAID webpage. You see there are already 2 million submissions of it. I will first give you some specific examples of some of the tools that are available before we go to looking at what are the exact tools and algorithms, and then also some practice of it, so that you get some hands-on experience. So the first genomes that were published from the China CDC was early on in January 2020. And you can see that since then, already now more than 2 million genomes have been shared from 188 countries. You also see that this platform, GISAID, has a unique sharing mechanism that allows to protect the submitter. So this protects also submitters that do not have the bandwidth or the expertise to immediately make use of the sequence, like make a paper. So if you submit to a public domain archive, people can take your sequence, can make a patent, can make a paper. They don't need to acknowledge you at all. But in this GISAID database, you need to register yourself. And then you agree that if you want to make use of the sequence, you need to collaborate with the person who submitted the sequence. This allows two things. First of all, the one who made the sequence, the country, for example, one of our ASEAN countries, is able to share sequences with other countries without the risk that someone else takes the information and makes the paper without them. And that's a very nice system that, as you can see, resulted in a very big difference in terms of where the sequences are found in this outbreak. So high performance is not a question of necessarily just who has the bigger computer, but also who has the better sharing mechanism. And transparent sharing, where you acknowledge the submitter, is a very important aspect of this GISAID database. And our group in Singapore, for example, is also contributing to some of the curation of the work. So when sequences come in, we help looking at the quality of the sequences, make sure they are fine. But this is a global effort. So we also have colleagues in South America that do this when we sleep here in Asia. And we also had colleagues in Europe and North America and in Australia helping us. And what you see here is if you just look at the last 30 days from when I took this screenshot, there are a lot of countries. Everything that is not white has submitted sequences within the last 30 days. And this is not just submitted within the last 30 days, but collected from a person, sequenced, and submitted. And this is the first time in a virus outbreak that we have access to real-time information. And this real-time information is very important because then we can apply a whole lot of tools to understand evolution of the virus. And this is just a list of some of them. So if you register to the GISAID webpage, you will see a lot more tools here. But also if you don't register, you just go to gisaid.org. You can already follow some the submission tracking that I showed to you. Our last exercise will be how can you read the phylogenetic tree and look at it? And this is constantly updated, and I will focus on the ASEAN region and the ASEAN tree. And of course, also of great interest, how can you follow variants and detect new variants and where do they go, like the Delta variant, for example. And just to give you some example of the tools that our bioinformaticians have developed for monitoring mutations. For example, this tool tells you, was there a mutation in a specific primer set? So the one of the earlier primer sets from the Charité in Berlin was developed early on. And it turns out that after a while, viruses have accumulated mutation that do happen within one of the region here in the forward region, the end of it. And you can see and map where these mutations occurred because we have this global sharing of the data through the system and in real time. An application that needs more of the high performance computing. So to give you some example, this here, we need an alignment of sequences and we need to make a sequence similarity search. So this is the underlying tool. We will come back to that we do. Here, we are talking about phylogenetic trees. So this is where we compare the genomes and the sequences with each other. And these genomes, they can be colored in different ways, but because we have so many sequences, it has become very difficult to make those trees and update them regularly. And this has become a high performance computing task. And I will show you through about half of the lecture about those trees, about different methods, how to create trees. I will give you tips in which methods work fast. So if you have many sequences, which methods you can use. And also if you have very specific questions that you need very accurate trees, including even understanding the time when an outbreak started or a specific group of viruses appeared first. You can do that with phylogenetics, and we will cover that later. And as I mentioned before, you can go so detailed that if you have a virus and you want to know what is its closest relative, for example, some genome here from Singapore that has the nearest neighbor in Indonesia. I think these were sequenced by the Eichmann Institute. In ASEAN, there are many very, very good success stories, how the countries reacted and responded. And of course, it's a difficult situation and cannot always be perfect. But because we have also a regional surveillance, we can see where is the nearest neighbor, where did it come from? And you can do this with, again, a BLAST sequence similarity search that we will cover later. But also you can try to plug your sequence into a tree and find your sequence in these bigger trees. And that's the second part that we'll be looking at. A part that we will not go so much into detail includes the 3D structure modeling. This is what will be covered by colleagues later on. How do you model and simulate the dynamics of a structure? The simple way that we are doing is you go from the sequence by comparing the sequence with the one where you know the structure, and you just highlight where the mutation is in the structure. without explicit modeling. It's more like a mapping exercise. And this is also important because you can give quite a lot of information of the site. Is it near the receptor binding site? So where the spike protein recognizes the human host cell. And this means this can affect how strong it can bind. This can affect transmissibility. This is why it is actually quite powerful to have this very quick mapping from the sequence to the structure. And you also are interested in where did the mutation occur. You can look at it in a tree, and you can look at what are the interaction in 3D. For example, is an antibody binding there? And this is when we are looking at if a new variant could affect the vaccine, we would see that actually the mutations occur within antibody binding interfaces. And last but not least, we need to connect it to the body of literature work that exists, and this information can be annotated. So all of us cannot know all the papers, cannot be experts in everything. So what a bioinformatics system like this can do is integrate the information and make it easily accessible for you. So you can make an informed decision, and it saves you a lot of time. If you would do all these steps here manually, it would take you several hours. If you run it through the program, essentially it takes a query sequence, it makes a blast search to find the closest relatives, it makes an alignment, then it looks at the differences in the alignment, and then it just looks in a database at this position in the alignment in the reference. What do we know? And then you get a whole lot of information, and this only takes a second. So that's the power of bioinformatics in providing the information. And as mentioned, if you know that a mutation occurs more frequently and also it's in a relevant position, for example, antibody spine, you can start to predict which one is a relevant variant. And this is what we're constantly doing and monitoring. And together with the WHO, Public Health England, and several other agencies, people around the world are looking at what is the new variant coming up. And on the front page in GISAID.org variants, we summarize. We are part of the committee that gave the names to the variants, the alpha, beta, gamma, delta. And you can follow and trace where did it occur relative to the region, etc. there. So what you have seen here is how useful bioinformatics can be, how useful you can extract information from sequences. Several of these applications now, because of the volume number of sequences, already require high performance computing. So what I want to do now, first I will stop and allow you to ask some questions in the chat. But what I will go on doing is I will teach you some of these tools so that depending on the application area, you may find very useful outcomes and relevant results. Of course, in the case of this virus, it's easy to say that it really has been very relevant. And a lot of people are asking me if I have staff, if I have people. And it's a quite of an interesting job to do. And I hope many of you can get interested. And you can also do that in your countries. And I hope you can learn a few things here. So I will stop now briefly for some first questions in the chat. Or not just in the chat, just raise your hand and okay, I see two. I don't know who was first. I will start with the tech colleague Varavan, please.
Oh, thank you. I'm just wondering, there was the slide that you say take a second to accumulate the data about the vaccines and the mutation that can affect vaccines. I'm just curious that when you say it take a second, it means the whole set of computation in every aspect, or is that only some part of the computation that will required when we have a new variant of virus appeared? something like that?
Yeah, that's a good question. So what the program does, so this is also developed by my group, is called the COF server. And the first step is a blast search. So it starts with a query sequence against a reference. And because it's not against many sequences, it just takes a second. Once the blast alignment is generated, we know the position of the changes, and then everything else that you see is pre-calculated. So this is one of the tricks that we use, that we do not have to calculate everything from scratch. So we have a database of all this information pre-calculated, and we just use BLAST as a quick connection. So that takes 1 second for one sequence.
Okay.
Yeah.
Okay, thank you.
Very good question. Yeah. Nisha.
Yeah, good morning. My question is regarding the efficiency of vaccine. So we know right now all over the world, especially in Malaysia, vaccination program has got a lot of opinions. People are thinking that vaccination is going to bring about adverse effects, and it's actually posing fear. So my question is, bioinformatics for evaluating efficiency of vaccine is upon administration, or is after the development of vaccine itself? This is evaluating the product, or is actually evaluating the product upon application to the people?
I think, of course, ideally we would want to do everything, from safety to the knowing how every person responds to the vaccine and the virus. The part that I was referring to is mostly about looking at the virus itself. So did the virus change? Because when you're naturally infected or when you're vaccinated, you're being exposed to this spike protein, which is on the surface of the virus. And then you will develop antibodies that bind to it, like here in this case. And not everyone is creating the same antibodies. So that's not for everyone it works in the same way. But the part that we are looking at is, how did the virus change? So if the new version, like the Delta variant, has mutations where antibodies bind, that means maybe the antibodies that we have from the vaccine or from an earlier infection may not be able to bind so well anymore. And this is, in fact, true. You can predict that from the computational analysis. However, it's very important to say that the vaccine efficacy before to the normal unmutated virus is above 90% for several of the vaccines. So it's very high. And then when you have a variant that has multiple mutation, not just one in sites where antibodies bind, it's reduced to maybe 80%. And that means it's just a slight reduction. And this 80% means that you're still protected from severe disease. This includes also younger people. So there are a lot of studies that computationally we can predict some trends and some guidance, but you need to measure it in the real world for making the public health decisions. And in different countries where you have both vaccination as well as the variants, you can see that there is a big advantage of vaccinating. And so people are being protected from severe disease in all ages, not just the elderly, but also middle age, and in some countries also kids are already being vaccinated. And of course, it's everyone's decision, but from the scientific point of view and what we see in terms of how the vaccines work in this virus and how the virus evolves, vaccination really is a very powerful tool to allow all of us to go back to a better normal and be safer overall. That is also my opinion, of course. Tira-san, please.
Thank you for your talk, but actually I'm not very common within this field, but one of my question is whether you can predict that which variant will be give the severe disease to the human or not when you try to analyze this data.
Yeah, so it's not 100%. A lot of the things we're doing is predictive, so we'll be able to have some good guess. The mutation that I'm showing here is in the 3D structure, in the place where it recognizes the antibody. Then in the literature, we have found a colleague in Seattle has made an experiment where he showed that if you have this mutation, it binds better. So if it binds better, it can, but not always does, translate into higher transmissibility. And there are some of these mutations, like this one, or the N501Y, that's the alpha, beta, and gamma variants had, that allow the virus to infect better. So computationally, we were able to see that, but it's I wouldn't trust it just based on the prediction. And especially, we have to be very careful in not coming to early conclusions, because there is a lot of attention on this topic. So if you write a paper, you say you find a mutation that does this, and you don't have experimental evidence, I wouldn't necessarily believe it. And but at the same time, we know that you can partially predict by information transfer from the position in the structure and literature that it could bind better. It's a good question. I see another question in the chat. If you have information about the vaccine distribution in a country, can you use the tool to relate your results? You can compare some things. The different vaccines that are being made, they're all still based on the same spike protein mostly. So it doesn't really matter the type of vaccine you have. But if you're also talking about, let's say, the percentage of people being vaccinated, that may may be an interesting aspect, because the more people are vaccinated or have been infected previously, the more likely you will see escape mutations. So these escape mutations are those that happen where the antibody binds. And so this kind of correlation does exist, and you can use these tools to monitor. And this may result in, we have this experience from influenza, where every year colleagues come together and decide the strain, the virus has drifted, so we need a new updated vaccine. And maybe updated vaccine, we don't know yet, but with boosters, maybe in a year, will be what will be the reality also for this virus. And you're right, looking at vaccine distribution and the evolution of the virus together may be able to help guiding some of these decisions. Joshua, so I'll last question before I move on. Hi, Joshua.
Hi, good morning. Did I understand correctly that you use BLAST for your phylogenetic tree setup? You don't use multiple sequence alignment tools? So
we use multiple different approaches. Because now we are at the stage where we have 2 million sequences. We actually always create a multiple sequence alignment daily. I will come back to that later, and it takes almost a whole day to calculate it. But in this specific tool that I'm showing here, we need a fast result, and we only need to compare to the original reference, which is our anchor, where we have all the other information tagged to. So BLAST is sufficient here for doing that. And this tool is also a pipeline for annotation, and for the annotation pipeline, we have an additional multiple sequence alignment based approach that we do to verify, especially when the deletions, insertions. I see one more question, Amy.
Hello. I just wondering, like, after we BLAST 2 sequence, and then I want to know, like, how much minimum and maximum similarity between 2 sequence is acceptable to say, like, this sequence belongs to this variant or something like that.
Yeah. So it's a good question. First of all, you can think of it in sequence analysis in general. I will show you examples there too. When a sequence is similar, often it means the sequences are homologous, so they are evolutionary related, and they are likely to have the same structure, 3D structure. And the threshold in percent identity, where you can say this reliably, is in the range of 40%. However, I have already predicted correctly structures that were only 12% identical. And then instead of BLAST, you use different tools that use multiple sequence alignment information. For the variants themselves, the WHO definition is based on both a phylogenetic approach, which means it's not just the percent identity, but it's the relative grouping that you can find, as well as the existence of these marker mutations. For example, there are always this marker mutation that we know that are near the receptor on the antibody. So if you have both the correct position in the tree and the positions, marker mutations, then you would call it, it's this variant. I see that there are more questions. I'm happy to answer them. I will go faster through the rest. Femi, please.
Thank you, sir. We know that a lot of or millions points that we get from database. So when we design a vaccine to the in silico approach, how we decide to know or we decide to the concept again to make a good vaccine to molecular docking or in silico approach. Thank you, sir.
Okay, so I'm trying to summarize the question because the audio was a bit low. So you're talking when we have a lot of sequences, like 2 million sequences, and you want to do some docking studies, and how to choose which one to use. So I think this is a very good example because the docking method itself is a very time compute intense task. So you cannot do it for the 2 million sequences. So what I would suggest in this case, use an intermediate fast screen approach like we have here. For example, the information, you want to dock if a mutation affects the antibody binding. So you need a tool that goes quickly, that can scan the 2 million sequences and know that it's still within that distance of the antibody, that it's relevant for your docking study. And this is what our database is doing. So this tool, it can take one new sequence in, but we also constantly run it over all the genomes. So we actually have this information annotated of what amino acid mutations are there. So you can register at gizeh.org. You can go to the database and you can look at the mutations of interest. And then you can use a search filter to say, I want to know all the sequences that have this. Or for example, you pick your Indonesian sequences and you want to know, is there a new mutation that eventually could be in this position? You first use this tool to find this is one that has the mutation, and then you can make the 3D model of it and you can take it on for docking. So I see a general question here from the webinar. So our purpose is to talk about high performance computing, and my media training suggests that I shouldn't go into opinions here. And it's clearly so that the vaccination itself doesn't drive necessary evolution of the virus, because you have still a lot of people that are not vaccinated. In the case of influenza, we have a big vaccination program, but it's still the advantage that the virus has in its own individual small microenvironment that allows it to be selected. So it's a bit of a race with and against the virus, but unfortunately, not so many things that we do are really affecting the evolution of the virus. And nature has a lot of tricks that it goes around to whatever we want to try to make it to change. What is clear is that vaccination saves a lot of lives, and that is, I think, something we should consider. I see more question about. I'll just answer the last two ones from Philippines and Malaysia. So assessing the quality of the data, we are actually using information, including from this tool. For example, is it a new and rare mutation that occurred, that never occurred? Is it a mutation that doesn't look like a natural mutation, so that it's just a sequencing artifact? And what we have is some percentage identity thresholds to known genomes. When in a sequencing, you're not sure what base is there, then you put an N. So it's the number of ambiguous bases. And usually we want 95% of the genome to be covered for it to be of reasonable quality. And for high quality, we would even want 98%. So this genome has 30,000 bases. And it means if you have 98% defined, you still a lot of ends in there. And we use the information. It's like a knowledge transfer and a learning-based approach. If mutation or a difference like insertion deletion occurs more often, then it is more likely also to be true and not just an artifact. And we can do control steps if there is a correlation with a mutation, insertion, deletion, with only one lab or with only one method to be used. For example, Illumina versus nanopore. So we do all these things to make sure there is no bias in the data. And it's a very nice role for bioinformatics to make sure this works well. And there are many colleagues also around the world that look at the data and that have ideas in how to look at it. Last question here, Citi. So how do biologists validate the bioinformatics findings? For example, the structure. So if we come up with this tool to say that this mutation could affect the receptor binding, we don't necessarily want or need the 3D crystal structure of it, which is also one of the ways how you could actually verify that this is the case. What is done with colleagues, for example, in Singapore, but also in England, we predict these mutations to them, and they have a biological pseudovirus assay. So they have this protein, and they have a particle that is not a virus, but it's coated with this on the surface. And then they do the physical measurement. Is there an increase in binding or not? And this is one of the biological tests how they're doing. Or if you would predict a change in vaccine efficacy, our colleagues, for example, have a database of plasma from people that were either vaccinated or infected. And they test against the blood, they test the antibodies, if these antibodies still can recognize that the mutated version. So they compare the wild type versus the mutated version. And it's, the job of a bioinformatician often is very difficult. If you just start in a lab and you don't have a collaborator, it's very difficult to find one and convince them to do the work for you. So the best thing is if you already, at any time that you can, see what the experimental colleagues near you, what are the things that they need, and then you try to predict something for them. And they may want to, they learn that actually, oh, it's useful what you're predicting for them. And so that you create this collaboration that you can have both prediction as a fast early step and then the biological verification later. Okay, I think we have to move on. I'm very happy to answer all these questions, and I spend more time on this because I think it's on all of our minds, and it's very good and important that we cover that. So, but what I'm telling you about is sequence analysis in general can be very important. This is what I had described to you before, how a single change in the genome here could translate to a change in the protein, which can translate to a change in the structure. And this structure, if it has the function, for example, to recognize specific parts in the cell, then can change how the virus behaves. This can change the severity. Like I think, Tiro-san, maybe you asked that question. So if there's a mutation here that allows the virus to infect cells that are deeper down in the lung, for influenza, we can do that. And we know that the virus could be more severe. But because the infection is deeper down in the lung and not in the upper respiratory tract, it's also not so easy to spread because it stays in the patient, it has severe symptoms and stays there. Whereas another virus like the H1N1 pandemic, like the swine flu, it infected mostly the upper respiratory tract, and that's why it became a pandemic, because it could be undetected for a while and it was spreading easily. So this is how you go from the sequence to the structure to the mechanism. And the same concept that we're using here for viruses is also not limited to viruses. So always think of it, you have sequence, we know we can go to the structure quickly, and we can also compare the sequence like in a phylogenetic trees. So just to give you a very quick roundup, we are working also with companies where we're looking at enzymes that create molecules, and we are optimizing enzymes. We are finding natural enzymes that can produce certain flavors naturally, for example. And that's one of the aspects that you can do here. And it's not just for flavors in food like vanillin, but also for drugs. If you want to make drugs using enzymes, you can predict which sequence will maybe do the reaction for you. And in another question here, you can also look at the safety of some of the proteins. You know, our food is full of proteins. If you have a shampoo that has some plant materials full of proteins, some of the proteins, like peanut, can cause allergy. And if by using these similarity tools, both at the sequence and at the structure level, you can predict how similar is the protein to a known allergen. And through that, you can predict partially the risk of it. And this is also useful for product development, but also for safety assessment. If these new alternative proteins, like this Impossible Burger, for example, if they use plant proteins that are normal, but you need to know and make sure that they are not similar to any known allergen. Like peanut is also a plant, so you want to make sure it's far enough away from it. And again, bioinformatics can help you guiding that, but you need some additional validation. And of course, very important, we are in the age of genomics, and precision medicine is important. So if we have a variant here, then the mutation can affect the binding of drugs, for example. So not all drugs work equally for all people, and precision medicine heavily uses this ability of bioinformatics to go from sequence to structure very quickly. So you've seen there are a lot of things that you can do by sequence analysis, and I want you to take home 3 key concepts in sequence analysis. First of all, that the sequence similarity, like we find out with BLAST or multiple sequence alignment, can give clues to the ancestry. So where did it come from, or are they related? Allows to trace back history and the origin of sequences. However, at the same time, we also need to be aware that sequence similarity can appear by chance. And this is then the question of that came up with the presented entity. What presented entity, or what are the reasons or factors that allow us to distinguish if a sequence occurs by chance, if a similarity occurs by chance or not? And last but not least, if you go from the sequence to the structure and using evolutionary information, we can elaborate on some of the functions that the residues do, like the receptor binding, the antibody binding. And all this together is important for sequence analysis. And this is what we often do in my group. And this is an old paper that we did just in the beginning of the swine flu. It took only 1 week of work, but it was viewed in 105 countries within the last year. And because computationally we are able to work fast and characterize the new virus very easily, it receives a lot of attention. And the part that I will cover in our lecture here is this concept of how a protein is separated in different functional parts, and how to make a sequence alignment, what are the common tools, and as well as how to make a phylogenetic tree and what are the common tools. So these are the take-home message I want to give to you. And the other part will be covered by my other colleagues that have very, very interesting parts on the structure. So I want to start with the segmentation of the protein sequence. So a protein is not just, of course, a linear sequence, but it forms into a 3D shape. But it is not just one big ball. It's actually consistent of multiple different domains and different regions. Sometimes they are structured, and we call them globular domains. Sometimes they are not structured, non-globular, or they have a very simple pattern, so that they are like linkers. So you can think of it like a ball with a linker. And it's important for you to understand. The reason why I'm telling you this is, if you do sequence similarity search, it works well in the globular domains. It does not work well for the non-globular regions. In this region, if you compare, you can get random hits, random similarity. And this is why this concept is important. And this is a better, simpler analogy. You can think of it like that. So you have the protein fold, the structure is like the leaf part. Then you have the stem, it's like the termini, like the tangling ends of the protein. If you compare the leaves, you can see they are very specific. So you can see some that are similar and some that are not similar. And then the result that you get with BLAST or with multiple sequence alignment makes sense. However, if you isolate and just look at the stem, they look pretty similar, and you wouldn't be able to tell from this that this actually has this shape or that it has this function. So that's why it's important to understand the domain architecture of a protein, that there are specific parts, the globular parts, and the unspecific parts. And again here, so you have a protein composed of multiple regions, the specific part, the globular domains, and the non-globular ones. And just one example that I was showing to you before, if you do assessment of allergenicity potential, like for a novel food, the old rules by the World Food Organization, Food and Agriculture Organization, was that if you have a small hexamer identical, there is already a risk of similarity. But the number of known allergens is so big that if you apply this rule to all of the human proteins, 60% of them would be classified as allergens. So that means we would constantly be fighting against ourselves, which of course is not true. And this is where I want to raise your awareness that similarity depends a lot on the statistics. There are some motifs that can occur more easily by chance, and others that are more specific. So again, same thing. You need to know if you have a specific or you have an unspecific feature. And this leads us to our first exercise, where I want you to go to that web page. I will first go through some example, and I will then give you some time to play around with it. In this website, ProSite, you can put in the sequence motif and search in the database how often it occurs. So sequence motif, you know, we have amino acids that are 20 different letters. It's not all the letters of the alphabet, but interestingly, you can also find normal English words or words in your language in the database. And it's not that there is any meaning to it in it being there. And for example, you can find the word health in sequences for which the structure is known. And it's, of course, it doesn't have the same meaning, but it tells you what are the statistics of finding things. And what I want to show you is how often can you find this word, how often can you find this, and this, and why. So first of all, health, at that time when I made those slides, it may have changed by now, you would only find it once. This is because we have a long set of letters, and we also have complicated mix of letters. We call this a complex sequence. So if we make it shorter from health to just the heel without the TH, so this the TH here you need to forget. No, sorry, that's actually okay. So what I did here in this was, if I search in the structure database with 100,000 sequences, I would only get one hit. If I search in a bigger database with 100 million sequences, you get 500 hits. So that's one of the notions to get. The probability of finding a motif depends on the database size that you're searching. So the big database, you will find more hits of the same. Now coming back to what I thought I was mentioning. So you have a long word with one hit, then you shorten it, you remove the TH, and you get 1,000 hits. This is because it's statistically much easier to find just these small 4 letters. And then what happens if you go further down with this, you change the H to an A. So for this, you need to know that H-histidine is actually a rare amino acid. That means it's not so common to occur. So if you replace it with a more common amino acid, you will find more often. And if you do this and make a simple motif, a simple repetitive motif, you will find the most hits. And these are very important concepts for you to understand how random hits and the insignificant sequence similarity come about. So if anyone tells me that, this virus has this AAAL motif, then I don't think it is very relevant, because I know it's just a very common motif that occurs by chance in anything that is also not related to viruses. So the functional significance of it may be much lower. And this is based on the statistics of the length of the word, the size of the database, as well as the frequency of the letter that are in that word in the database. And this brings us to our first exercise. And because of time, I will give you about 10 minutes. I want you to go to this web page, and I want you to find the longest real word that you can think of that has at least one hit in the PDB database. I will go back here to the starting page. You have the link here on top, and you need to choose the option 2. Then you type in your motive here, you select the PDB database, and you click Start the Scan. So you think of a word that you think has only amino acid letters and could occur in the database. And I've done this many years with different types of students. Thanks for posting the link in the chat. That's very useful. And if you find a word that is longer, so right now we have health, for example, 6 letters. So if anyone finds another word with 6 letters, post it in the chat. And if you find a word that is even bigger than 6 letters, please also post it in the chat. And again, those that manage to find some words, I will take note of the names and pass them on to the colleagues. Amy, was it in the sequence? Because it could be true. Of course, this is the name of the organism, but sometimes people make mistakes and they submit to the database. So that means the name of the organism has been posted in the sequence. So I see we are overloading the server. How many are we in the? Because I've done that already also with like 40, 50 people. I think we need to be a bit patient. So Amy, can you tell the code of where you found the hit? Because I've never seen that one. I know sometimes those things happen as a mistake, but that would be really the longest. Yeah, I forgot. We also have a whole lot more from the webinar there. But it doesn't matter. I think you can play around with it also a bit. I'm curious. I will look up the example from Amy. But it's actually the name of the organism. So in a way, it is a word. And sometimes we see also in the virus sequences when you do the quality check that we see the name of a country or city in the sequence. And then we know it is actually, something went wrong with the bioinformatics pipeline that put the country name together in the sequence. And in this case, this looks like the organism.
There is no hit, sir.
Okay. It can happen nevertheless. So I don't. Is anyone from the students? Do you have any result so far? I can tell you otherwise. I think what was the most common one that we have found in other trials of the same. Yeah, I see that the problem is you cannot make a systematic scan. So if once you manage to get a result, but you don't get any hit, then you need to start again, and then it may not work again, and the server's overloaded. So I'm trying to remember what were the longer ones that we had. I think was just in the level of 8 that that we got, and one of those that only used the natural 20 amino acid letters was allergen, so 8 letters, and so imagine you have a protein that naturally has the name allergen written in it, but fair enough, it is not allergenic, or at least it doesn't look like to be. The reason for this exercise again was to make you aware that when we do sequence similarity searches, there can be false positive hits. And we need to know what are the factors influencing that. I already showed to you with the leaf and the stem example that there are low complexity regions that are potentially causing this. And the complexity, for example, the number of different letters or how common they are, so as the length of the similarity stretch are important. And I think that's the take-home message from this exercise. And I think I'll have to redesign this depending on the number of people doing it. When we do it in person, we are doing it in the range of up to 40 people or so, and it still worked. But a lot more maybe doing this and trying now. So we should actually tell our colleagues in Europe, in this case in Switzerland, they should have a scalable high performance computing environment to deal with the games that some of the researchers throw at them. So just to recap, what I was trying to do is to give you the example that a protein has multiple different regions. Some of them have the 3D structure, some of them don't have. And this is a summary of what are the non-globular regions. For example, a signal peptide, a transmembrane region are non-globular. These are helical, so they do have a secondary structure. But all these are regions where if you have a similarity only in that region, it's not necessary because of ancestry. The same thing with these polar low complexity regions. The tool to calculate this is called ZEG. And the tools that I'm mentioning here are a good tip to look at or keep in mind for the exam question. So ZEG is one of the tools that allows it to detect this low complexity region. Like I gave the example with the motif like AAAL. So that is a low complexity region. That means it's not many different amino acids. And if you have a hit only in that region, that means it's not necessarily significant. Similar called coils, with the program coils, they also tell you a lot of repeats, for example, of E's, E, that are part of a helix. So again, here, if you have similarity, it can be for many different reasons. And when you do this analysis, the parts that are not structured, they can still be functionally important. There could be a modification like a phosphorylation or a lipid anchor. And that's why we also don't just cut them off and disregard them, but we need to analyze them. And then last but not least, we have the known domains that we are comparing to. And then everything else that we don't know the structure of, that we cannot predict, could be new domains. And we know that they are non-globular, so they are relevant. And you could calculate some similarity there. So if you were not familiar with protein sequences before, keep in mind that they are not just 3D structure, but they also have ends and sometimes consist of multiple motifs. And this is what happens to a protein in the lifetime of itself. It's undergoing multiple modifications. And for example, here, this is an enzyme that gets this lipid and puts it onto the red protein. Because it has this a lipid attached, it goes to the membrane. But you have additional things happening. For example, you have another enzyme cutting off this tail to make it even easier to go to the membrane, or you can have another protein interacting with it. So this story of all the different motifs together is what makes up the function of the protein. And I will not spend much time on it. As a bioinformatician, when you describe that, you can try to rationalize why the mechanism of the protein relates to specific features, like the membrane anchor that allows it to go to the membrane. And those slides are available to you for download. You can read it more carefully. And this is a good way of analyzing the function of protein. But I will not ask exactly this as the quiz question. But I think it's good for you to know. So if you're interested, please go back to the slides and take a look at some of the examples. And this is how in the UniProt database, you can have these different features of a protein annotated. And you can have, when you know where a mutation is, from the linear sequence and the domain architecture, already have some idea. And this is also how the COF server that I described works. So you have a database of the annotation of the position. And then if by sequence similarity, you see there is a mutation and you say, oh, it's in this feature. So this feature could be the receptor binding site or the antibody binding site that is already annotated here in the one-dimensional space. And this was the first quick part. Now we're moving on to the multiple sequence alignments that, of course, we cannot do a whole lot of detailed on this in the lecture, but I think it's important for you to know that the concept that you've learned before is for homologous sequences. So it's for the leaf part of the domain, the 3D structure domain. The part that are non-structured, that are linkers, you will get false positive hits. And that's what was the purpose of the first part. So if you know that, and then it helps you evaluating, is my similarity significant? And this is what you do, for example, with the BLAST tool. The biggest database for all proteins, for all organisms in that case would be the NR database from NCBI. And you would, for example, turn on a low complexity filter that masks out the regions that you know could cause false positives. And if you need to find more detail, then you can do HH-PRED, for example, or the Google AlphaFold, or you could go to databases that have already annotated the different relationships, the similarities. They group them together. So going to the BLAST search. So BLAST stands for the Basic Local Alignment Search Tool. And that's one of the most commonly used tools, and it's still useful now. There are, of course, improvements that one can do. And I want you to understand from what we have looked at before how it works. So first of all, it looks in the index database, and it starts with a very small peptide, the 3 amino acids. So you've seen that when you have this short motif, like for the health example, and then you reduce it to heal, then you start to see hits. So what BLAST does essentially is it takes from your query sequence the small letters and searches in the database just for those. So there will be many unspecific hits still, but instead of the whole database of, for example, several billion or 100 million sequences that are now in NR, so it's, I think, 500 million already, then it will only give you back 10,000 candidates. Many of the 10,000 will also be wrong still. But then the next step is that from the hit, from the 3 letters that are shared, it will extend to the site to create a local alignment, and it will score how long is my similarity. So it uses the 3 amino acid short pattern as an anchor to find possible candidates, and then it extends the motif on the site to let you know if there is a significant similarity. And the significance of the similarity is done by scoring identical residues as well as similar residues, based on a substitution matrix called Blossom 62. And this score is translated as an extreme value distribution into an e-value, which gives you the probability of this hit being random. Like, how often would you expect the same score to occur in this database by chance? And that is the e-value threshold. And the modification that you can do to this is you can, for example, increase the seed pattern size so that you get much less hits to do the more detailed alignment. So these are ways how you can speed up. This is important for the high performance computing and understanding. And these are all the things that we use as tricks, depending on the question that we have, to speed up our searches. And then you can have different types of searches. For example, I showed to you proteins, and you can search protein against protein. You can search a nucleotide sequence against a nucleotide query. You can search a protein database against nucleotide, and that's complicated because you need to translate in between so that you meet at the same time. And in the same way, you can also search from a nucleotide database with a protein query. And on the BLAST webpage, you can find also some details on this. It's one of the most classical tools, and it's still very powerful. So that's why I think I will never stop teaching that, because it's still very useful. And then there are specialized ones that, for example, build profiles that put more sequences together. Instead of a single sequence, you get evolutionary information included. And this evolutionary information, I will look at with you for the alignment. That's the very same thing that we are looking at. So if alignment of homologous sequences, my preferred tool here is from MAFT. That was developed by Japanese colleagues. There are many other tools that I will also show you, give you some examples. But they have a very nice way of getting a very good alignment relatively fast. And this is how an alignment works. So imagine we have these 4 sequences. Of course, this is just a short sequence from human, rice, fly, and mouse. And what we need to do is to align them, is we slide them against each other. And until we find the alignment, the position that gives the minimal pairwise distance. So in computer science, this is called the Hamming distance. And so if we are sliding the mouse sequence here along human, we see the best alignment is when there is only one difference, when they are like this. So that's quite simple. So then we do the same for the other organisms, human, rice, human, fly. And what we get is a matrix, a pairwise similarity matrix, or identity matrix in this case. So we know that the sequences to themselves are identical, and then this is the distance that we have here. And in order to create a multiple sequence alignment, this step is actually very similar to steps that are used by the phylogenetic trees. So that's why we can later use the same concept. So we start with the most similar pair, and then which was the human and mouse with only one difference. And then we have this, and we slide the next sequence through it, the fly. And again, in the simple example, of course, with the same lengths, you see it comes in nicely. Although there are differences, it's still similar. And also again with the rice. And these are the steps that one of the popular programs, Cluster W, does as one of the first steps. And then other tools are using some speed ups to get there faster. But that's all that is to it. So you want to compare the sequences and want to minimize the distances between them. And this is what the multiple sequence alignments are doing. And Cluster W is a bit like BLAST in terms of it was very widely used. But there are now better evolutions of it, like Cluster Omega is the latest version of it. Then these progressive tools, they are starting to add one sequence at a time. And then you have those tools here that also start like that, but they do iterative refinement. So after you get the first alignment, both muscle and MAFT, they are redoing some of the steps. And in the case of MAFT, the first step of getting an alignment similarity is based on hexamer matches, so a k-mer score, which further speeds up the process so that you get the first distance matrix based on longer words. Just like when you do the blast search with longer words, you will get less false positive hits. MAFT uses that trick internally. And then there are also probabilistic methods, and the great detail of that is not so important. But you can always get it to more extreme. The most extreme is you have the whole pairwise distance matrix, and you do backtracking via dynamic programming, which would give you an exact solution. So if you're interested in doing that, there is a book, a classical book by Richard Durbin on sequence analysis that also teaches the dynamic programming algorithm. However, for most current problems, I think this is not realistic, and it takes quite long. So what we want to do is get some idea what are the different programs. Again, just like in the blast search, you can go from a single sequence to multiple sequences to have more information included, which is this profile guided and profile to profile alignments. And you can even include some structural information. So if there is a loop or if there is a secondary structure that you don't want to break, you can encode this information that will help improving your alignment. And I think it's been a quite of a stable field over the last 10 years, but 20 years ago, many of these tools just started to come up. So there haven't been very big jumps since these tools have become available. And there are smaller improvements happening all the time. And I want to give you some quick example. I need to make sure with time, we have about 1 hour left, so I think I have to move a bit faster through that. I want to just give you this one example here. For this alignment, which of them to you looks more conserved? You know that, of course, the colors are being used to indicate some level of the conservation. I see a lot of Bs coming in, very nice. Yes, the B is more conserved. And although you see colors here, the first one is actually a random alignment from proteins that I had together in another exercise for students. And this is to show you that even random sequences sometimes can have similarity that looks like similarity. And it comes from these statistics. Small motifs that can be partially shared, like this EAQ, then it looks like, oh, this must be very conserved. And an algorithm, the multiple sequence design algorithm, will always try to give you the best answer, but doesn't know if it actually makes sense. So you give it wrong sequences, even protein and nucleotide sequences, you will you will get back an alignment, but it's not necessarily always true. So that's why you need to make sure that you only align sequences that you know are related, either by the sequence similarity search like BLAST, with the e-value and the similarity not being in the non-globular low complexity regions. And then you know that it's also some other patterns like hydrophobic residues here that are important. And this is the warning that I was just mentioning. An alignment program will tell you whatever you give as an input. The fact that it gives you an output doesn't mean that the alignment is correct. So it's up to you to look at it. And one way of looking at it that I recommend is this tool by a colleague called Geoff Barton, ChalView. It's, of course, the free tool. And you can see alignments, you can make alignments, you can edit alignments, and you can see multiple features, including like a small simple tree, but also the structure to it. So I will not go into great detail, but I want to mention this to you because it may be useful to you. So when I say that you can judge how good an alignment is by looking at it, we know in the first example, when I give the when I said hydrophobic residues want to be in the inside and polar residues on the outside. The same pattern you can see here in the coloring, and this coloring used here is actually the original protein coloring from cluster X and cluster W, and that many people use, including myself, for the coloring schemes. And you see that you have a hydrophobic pattern here. And you also have a hydrophobic pattern here, and that is with some distances. And the conservation of the hydrophobic pattern is actually more important to know that there is a similar fold, even though the sequences are very remotely related only. And what we need to consider here is, I don't know all your background, but that's a very useful consideration. When you look at sequences and sequence alignments, we need to know what are the properties of the different residues. For example, if you know that these letters are aromatic and they are very hydrophobic, or if you know that others are polar, that's why if you want to interpret the protein alignment, you need to know some of these properties. And of course, there are charts that help you with doing that. And you can also group them nicely. If you don't know this by heart, this is a very useful tool. So how you can analyze and judge alignments. And of course, also if the structure is known or if known motifs are there, you will want an alignment where the conserved secondary structure elements are there and the loop regions, there's where you can have gaps, for example. So you can learn from the structure also if your alignment is correct, and vice versa. You can learn how to predict the structure based on the alignment. And last but not least, if you have residues that you know that are important for the function of the protein, you will want them to be conserved. And here is another example. Similar, please type in the chat which one do you think is the best of this. I will take note of your names in the chat. Yep, I see a lot of the math. And does any one of you know why I think this is the best in this case? Yeah, I see Johannes wrote the histidine. Yeah, yeah. Yes, very good. So these are still the globins. And in this alignment, we know they are structurally related. And the histidine is actually very important. It's difficult to align for the methods because there are some gaps around. But this histidine is actually the one binding to the heme group. And this is where, of course, the globins, they carry oxygen, for example, in our blood. And there is an iron and the heme in the middle of it that is important. And the histidine is the one exactly binding there to the middle of it. And this is why it is important that the histidines are well aligned here so that you can characteristically say, yeah, it has this function. So, and I will not go into detail here, but using ChalView, for example, you can get some idea about the residue, or the role of the residue. Is it conserved and hydrophobic in the middle, important for the fold, or is it not conserved? You need to know again what are the properties of the amino acids, like a big residue, small residue, polar. And then you know that the conserved hydrophobic residue pointing into the middle, like here, is important for the fold. A hydrophobic residue conserved on the outside may be important for protein interaction. And because that's not favorable, as you know from the score. At the same time, if you have a residue that is not conserved, like this one here, and on the surface, it's just a random loop. So it doesn't really matter so much if this one is changed. And if you apply the same thing, for example, to viruses, you can see how common the virus mutates relative to specific functional sites. In this case, this was an influenza neuraminidase with Tamiflu bound. And we looked at all the globally circulating sequences and counted how often mutations occur near the drug binding site. And fair enough, the known drug-resistant mutation occurred the most often, this mutation here. But we also found other additional mutations that were not known yet to be drug resistance mutations. So we predicted this to colleagues, and they experimentally verified it, and they could actually see it. And this all comes from going from the sequence to the structure. And I think we have about 40 minutes left. Then I want to give you one exercise where we do sequence alignment and the blast search. I think we may not do it fully, but the example I can give to you, maybe I'll ask you just to follow me and not do it yourself, so that we don't overload the servers, and you can walk through it yourself. So what I will do is I will share now the other screen. So the background of this exercise is that while the main labs in China have shared their genomes via the GIST platform, there was also one sequence that was shared in a blog a day later than the official sequences. But this sequence in the blog also made it into the American database, the public domain archive GenBank. So you see that the sequence has been updated, and you see that this is the 0.1 version. So this one became online here 13th of January, so three days after it. What I want to do now is I'll get the FASTA sequence. You see that's the genome with 30,000. Then, so I need to move this a bit away. Then I'm going for the MAF online server. So this, of course, if not everyone does it the same, they will also be overwhelmed. And I'll just paste the first sequence in. So MAF is the multiple sequence alignment tool that I had recommended. And now what I want to compare it with, so see the current version. So we have the original version that came from a blog, and we have now the current version that was fixed on 17th of January only. This is the 0.2 version. And this is the reference genome at the NCBI. Whereas if you go to the GIST database, you have a higher quality sequence for a longer time. So I'm pasting this here. So we're making a multiple sequence alignment only of two residues, of two genomes. I will use the default option here. It has accurate ways, and it has a switch depending on the size of the genome. I'm getting the same issue. Too busy. Let me see if I still have the result open somewhere else. Because of course I did try that before. So this is the result that you get when you do the multiple sequence alignment of this first version, the .1 and the .2. And one thing you see is that in the end of it, there is actually quite of a stretch that doesn't align well. And you can, it was interesting to see, so what is the difference between these two? And then what you can do is you take this part that is not the same, and you compare it and make a blast search. So I cut out this portion, and I go to the NCY BLAST page, and I search against the biggest database, and database from Genbank, to know is this another part of the virus? Sometimes the assembly didn't work and it mixes the front to the end. Or was it an animal that it came from? Because sometimes those things, if it's not looking very similar, maybe it was really pointing to the source. So the top hit here, so long story short, is that the top hit is actually a human DNA sequence. So this sequence that was shared via the blog was contaminated by the person who sequenced it, also had their own DNA amplified, and then so multiple mistakes, and the assembly program also added the human sequence into it to the end. So this is a very common artifact because in all the sample handling, it's almost unavoidable that you put some of your own DNA in there. But then in the library creation or so, and if you're careful, and in the bioinformatics algorithm, you can avoid that. So this is just to show that it's important to know when the quality of genomes are important, especially when it's a new pathogen, and there are reliable sources where you can get this information from. And of course, in the first few weeks, there was still a lot of confusion in what is the right genome. But if you look at the GISAID, where the official genomes from 3 different institutes, they found the same identical genomes came out, and as a consequence, also the public domain archive, where people just got the sequence submitted via a blog, that they changed it to represent what is the actual consensus of the high quality genomes. And in this little exercise, you can try to do it yourself, and please don't overinterpret the similarities. You know, it's a simple mistake that they had the assembly of a human part in there, and they very quickly fixed it. But essentially, it shows an application of how you can do the sequence alignment, and also how you can do a blast search. And that was for this example. And it also shows that the online servers are not ideal for our classes, because they may be overloaded. And the MAFT server, when I run it, I run it always locally. It's quite easy to install in Ubuntu, for example. You can get it with apt-get automatically installed. It also scales quite nicely. So it's quite easily available also to in high-performance computing environments. And then there are different options to it. So moving on to the last part of what I wanted to show you, which is about the phylogenetic trees and finding, for example, the relationship between the sequences. First of all, some basics. And as you know, life on Earth is evolving over millions of years, and a lot of the species that you see are closely or less closely related to each other. And by the modern genome technologies, we know and can classify them. In previous times, this was done by morphology, by bone morphology, etc. And I'm wondering, and I want to ask you, you can type in the chat, to which of these 3 organisms on the left is the blue whale most closely related? Is it the whale shark, the cow, or the elephant? And I have to say that also in old times, just based on the bone morphology, the 100 years ago, before genetics, people also knew the right answer already. Yeah, we've seen quite some different changes. Of course, intuitively, something big swimming in the water should be related, right? Or other intuitively, like the elephant, what's very big, the blue whale is really big. The truth is, genetically and morphologically, the cow is actually more closely related. And the missing links in between, some of them are known. You can imagine like a cow living near water, also like a hippopotamus, for example, is related to that too. So you have something that is a mammal related to cows that lives near the water and goes more and more into the water. So you have like a hippopotamus that still has legs, goes in and out of the water. Then you have things like manatees that they also eat grass. They eat the seagrass in the flat. They are not in the deep ocean, but they are near the water. And so with genetics confirmed, we actually know that blue whale is more closely related to the cow among these 3 different ones. And the elephant is interesting. It's another subgroup. There is actually another organism that is as small as this, that is very closely related to elephant genetically. So it's not just the size that matters. So you have something that is this small, being closely related to the elephant, and it's just the expression of genes and how the environment shaped how these come together that makes how the animal looks like in the end. And of course, there is a big time frame that evolution takes for this. So what do we need for a phylogenetic tree? Essentially, all the things that I just told you before. The first step is we need to identify sequences that are related to evolution. So that means we need to get significant similarity hits. That's why you need to know that, okay, the similarity is not just in a small motif that could occur by chance. Then you make a multiple alignment of the sequences, just as I showed you, and then you build the tree based on the alignment. And the good alignment is crucial for a good tree. So going back to the same example, you start with sequences, you make the alignment, just like I showed you before. You start, for example, with the closest pair. And this is, instead of just making the multiple sequence alignment, we already use this now to make a tree. So the closest pair here is already the first node coming together. Then the next close sequence is one relative further. So this is how we get this tree structure together. And finally, we are doing this. We get our complete tree. And these steps are the same steps that you take in the classical neighbor joining algorithm. This is a very fast algorithm co-developed by multiple colleagues. One of them was a Japanese called Nei. And so there, it's quite intuitive how you get to this tree with this method. It's based on distance and nearest neighbors. The good thing is with this method, it's very fast. And of course, there are many different ways of doing it. And one of the softwares that you could use is a package called Negra. And usually I recommend that to biologists because it has a graphical user interface, but it also has a command line interface. And it has implemented several of the algorithms. There are many different other packages that people want to try, and this is one that I recommend. So this distance-based tree calculation is, as I showed to you, you slide the sequences and you want to make sure that they are coming together and aligning well, and you minimize the distance. There are different algorithms for that. And some of the names of these algorithms, they may also be asked, not for you to type in, but in a multiple choice question. For example, the question could be, which of the following list of 6 different algorithms is not a phylogenetic algorithm? So I will use some of those actual names and add a one random fake one. And you need to find this. So this could be one of the questions, for example. And what I want to summarize is that we have distance-based methods like neighbor joining, minimum evolution, or inference methods that are looking at multiple trees at the same time, as well as some additional information. So maximum parsimony, for example, looks like that the steps taken from the beginning of the tree to go to all the steps before and later on is makes sense. So that means if you have a mutation that goes from one starting point, then you get a new mutation and a new mutation and a new mutation. And if this mutation is only found along this branch, that means it's consistent and it's the most parsimonious solution. But if that mutation is also found in other parts, then its parsimony score would go down. And similarly, you can do this not just with some markers, but with the overall likelihood of a tree. How likely is it for a tree to exist? So, for example, we could make different permutations of the tree, and you want to know how likely is the probability that this tree is right. And the last method that is the most accurate one is called the Bayesian method. And this does Monte Carlo simulations of different trees, many millions of trees. So this is a high performance computing task. And after creating all these random trees with some different starting criteria, it chooses among those which one is the one that makes most sense. So this can start with a maximum likelihood criterion. And then essentially, it also follows distributions. So that probability distributions that you can give some prior assumption how you think the evolution works. And then you want to be within your tree, well within that distribution. So for the distance measures, we can do the simple count, like the Hamming distance. So identical, not identical. So how many differences are there? And then we also have substitution matrices. And that could be based on different protein matrices like the Blossom 62. In this case, JTT is one for James Taylor Thornton, HKY for nucleotide, Tamurane for nucleotide. These are different distance measures. And the substitution matrices, they just mean that, for example, if you have a difference between two positive charges, they are still more similar, although they're not identical, but they have similar properties, so they are more likely to be okay. So it's not just a yes-no answer, identical or not, but it gives some information of how often those transitions happen statistically in evolutionary data sets. I'll give you this information, but this detail will not be asked. So if you're curious, you can follow up further going back to the slides that are being shared. Another aspect is that the evolutionary rate within an alignment and within a protein or a gene is not always the same. There are different speeds of evolution if you're on the protein surface, for example, because you're allowed to make much more changes without disrupting the structure, versus inside, the structure is very hydrophobic and needs to be folded, and you cannot have mutations happening there. And that means the rate of evolution inside the protein and outside of the protein is different. But many times when you make these assumptions, you would assume a uniform rate across the whole length. So this is a simplification. Whereas a more accurate description is that you have this gamma distribution, which means you have some highly conserved invariable positions and others that are much more variable. So you allow a range of different evolutionary rates to happen. And this is, again, a parameter choice that you can introduce in the phylogenetic model. So you can choose how do you count the similarity, like identity or similarity based on physical chemical properties. You can choose the rate. Do you want it to be the same across the whole length of the alignment or with some gamma distribution? And the last thing is you want to evaluate the reliability. And this is done with a process called bootstrapping. And you do this by introducing variations. For example, you remove one of the sequences and you see how does this affect the rest of the tree. Are the same branches still grouped together? And this is called bootstrapping. And besides removing one sequence here, you can also remove a column. Like, how does this affect? And then you get the bootstrapping score in the tree that tells you how reliable is this grouping. And the reliability of the grouping is important for your assumptions. And I will come back to those examples later. And just to give you some guidance, you can make a very fast tree with neighbor joining. The Poisson correction essentially is an identity measure with just a distribution correction. You can use uniform rates, which often is good enough as an approximation. And because this is fast, you can do also 1,000 bootstraps. If you want to get a more detailed tree, and for publication, usually you would go with something like a maximum likelihood tree, you do all these other advanced features, like the gamma distribution, you use a different distance, and then you get the result. But the most simple rule is that in many cases, this simple tree actually is giving you the same result. And this is a very good thing, because first of all, you can get quick results first, and then so that you know, you get a hypothesis of how do you calculate similarity. And if you're looking at the different, if you're looking at the maximum likelihood trees, you see the more accurate picture. But if you see the same picture, you know that you cannot make many mistakes in interpretation. That both neighbor joining and maximum likelihood are giving you similar results. And so this is an important rule for you to interpret. If you're getting different results with different methods, you should be careful with your interpretation. At the same time, if the fast and the thorough method give you similar results, you can also be quite confident that the relative grouping that you're discussing and that you're finding is correct. And last but not least, here at the bottom, you have a ranking of the methods of the different spectrum. So the Beast Bayesian method, that is the most accurate, but it's also the slowest. Then there are maximum likelihood methods. Actually, all those in the middle are maximum likelihood methods. And then there are fast methods. Here on this end, we have neighbor joining methods. Here we have the Bayesian method, and in the middle, we have maximum likelihood methods. Beast takes the longest. They also have some parallel implementation. So this you can do in a high performance computing. But here these methods, for example, PhiML is a classical method for maximum likelihood. Another colleague took the similar approach, made a faster method called RexML. And then he made it even faster in a method called RexML NG. And other colleagues developed another tool called IQ-tree that is even faster than it and still is giving maximum likelihood. And this is literally how, in the course of this pandemic, when there were just 100 genomes there, I was using these methods to start with, to make a tree, to make analysis. And the more genomes came in, the more we had to move to faster methods, like these ones over there. And often now we use a neighbor joining tree as a quick proxy. And another method that we're using is actually called rapid NJ, so for rapid neighbor joining. And but for more accurate trees, we use IQ-tree that you can do for a few hundred genomes, maybe for up to 1,000 genomes, but of course not to the level of 500,000. And that's where really both high performance as well as algorithmic design plays a big role. So bottom line, there are fast methods that are based on distance. Then there are thorough methods that do a proper statistical sampling of the space of the possible trees. Simple rule is, if the different methods give you a similar result, they are more likely to be correct, so you can be more confident in your interpretation. You have the Bayesian method that are accurate but slow. You have the maximum likelihood method that are in between. It is totally fine to make publications using IQ-tree, for example. Many of the SARS-CoV-2 papers are using that. And if you need make super big trees, you need even faster methods, and often they're purely based on neighbor joining distance or even just on maximum parsimony, in the sense of just using mutations, not the whole sequence for comparison. So this slide is just to show you what all can go wrong. So I will not go through it with you. Essentially, if you have a mistake in the alignment, your tree will be wrong too. And then there are many different things that could cause this mistake. So if you're interested in learning a bit more about phylogeny, go back to that slide and read through all the potential factors that can influence if your tree is wrong. Most often it's because the alignment itself is wrong. So I want to just give you one more example of the advanced phylogenetic methods from the Bayesian method and how it's being used. And we're talking here about the molecular clock analysis, because we know that viruses accumulate mutations over time. And this is so regular that we can actually use this to calculate like a molecular clock. And what we see in this example, this was in 2016, Singapore also had a Zika outbreak. This was following when there was a big outbreak in South America from the Zika virus in 2015, 2016. And of course, people were constantly surveilling if they also got the virus. And Zika, different from the respiratory virus, needs mosquito as a vector, but we do have them here in the region. And historically, this virus was first identified in 1947 in Uganda, was circulating still in Africa. And you see here that the whole African group is separate. Then in the 60s, the first sample was found in Malaysia. So it crossed over and was found in the region. If it's found first in one place, doesn't mean it's the first place it appeared in Southeast Asia, but this was the first place that detected the virus. So there is a sample in 1966 in Malaysia. And since then, it has been circulating endemically here in Southeast Asia until there were some outbreaks, bringing it to Micronesia in 2007 and French Polynesia in 2013. We know from the phylogeny that the outbreak in South America, in the Caribbean, was coming from this strain, from French Polynesia. So the travel direction of the virus was from Africa to Southeast Asia, to Polynesia, to South America and the Caribbean. And from there, it often went back. Because of travelers brought it, this was also the time, I think, of the World Cup and the Olympics were around this time. And you see that you could clearly see, this was one case in Singapore, but it looked like the strain found here. So it was a traveler that came back from Brazil. However, when Singapore had an outbreak of a few hundred cases in 2016, when you look at it in the tree, it actually did not look like it was coming from here. And it was more similar to the earlier viruses that have been circulating in the region. And what you can do here is now you test a molecular clock model using the Bayesian method. So you know the time of your sample, of your sequence here, and you take the genetic distance, just like we measured before. And you see there is a direct correlation between the genetic distance and the time. And this tells you that there is a valid molecular clock model in this case. And with this model, you can tell what is the time. When was the last common ancestor of the Brazilian outbreak strain and the strain that caused the outbreak in Singapore? And this was in 2010, 6 years ago. So this was way before that time. So it's more similar to other viruses that were circulating in the region. And again, this doesn't mean that the virus necessarily comes from there, but the country that has this sequenced has a good surveillance system so that they detect it. So in this case, we could say that the outbreak was not coming from South America, but rather from endemic viruses circulating within the region. And that's an interesting aspect. And if you're curious in how to get into doing this kind of analysis, Beast has a lot of tutorials that can bring you through. You first start with the starting tree that you create with the methods like I described. You check for the clock model, and then you're looking at how good is your sampling. That's the essentially when you make many millions of trees, 10 to 100 million trees, you need to wait, and then you need to judge. And a quality measure that this tool has is to give you this effective sample size, ESS. And you only need to check that this ESS is above 200 or above 150, then you know that your simulation converged. So these are Monte Carlo simulations of many million trees together, and you want convergence. And a measure of if it converged in this method is if this ESS is above 200. So this here, everything is fine. And I'll show you here an example where it is. So you see there are many that are going down below. So here what you need to do is you need to simulate more. And that means, of course, it takes longer time. And but there are two things you can do: simulate more, or also change some of your model assumptions. some of the distributions. So without going into much detail, you have these slides available. You can go through these examples. You can make some meaningful interpretations, and this is something that is very important. I'm also very happy to see there are many colleagues from Thailand among you. Several of you also asked some questions. And in the beginning of the outbreak, I also worked with one of your colleagues, Pillai Luk, and it was actually the Thailand team that first reported genomes, full genomes from outside of China in this outbreak, very early. These were travelers from Wuhan that visited Bangkok. Thailand already had thermal scanners there at the airport, so they were fished out because they had fever. And then they were confirmed with PCR that they had the virus. And Pile Luk's team sequenced it and looked at the genomes. And we helped, of course, sharing and also looking at the genomes. And you could see that the mutations were very few at the time. So the genomes of these first people in Thailand were the same as the ones from the typical cases that were in the seafood market in Wuhan at the time. So the virus hadn't changed much then. And I think this is a nice example and application how this kind of analysis helps in real world situation. And I have a similar example here in Singapore. When the first Wuhan tourists came into Singapore, there were different clusters that our Ministry of Health was investigating. So you have investigation by epidemiological teams so that they do contact tracing. They know, oh, this person went, for example, to that church. Or in this case, these were tourists that went into a Chinese health treatment shop. And we had the tour guide, you had staff working in the shop, and you had at her home the domestic helper, 6-month-old son, and a husband. And by looking at the genomes of the viruses that they got, you could reconstruct and confirm the contact network and the epidemiological picture. And this was one paper also published very early. It was already cited more than 500 times. It shows the power of combining phylogenetics with epidemiological information. And not always can we say for sure how the viruses are linked, because often they are very similar. For example, the cluster at home, these 3 cases, the helper, the 6-month-old baby, and the husband, their genome of the virus that they had, they were all identical. 30,000 bases, all identical. But to the wife in between and the tour guide and the tourists in Wuhan, there was always a step of 1 mutation away, and this was nicely compatible with the pattern. So this essentially would lead us to the last exercise, which I will go through with you also in a live demonstration. And just to summarize that with this exercise, we are done. I know you have a short lunch break, so I will definitely not overrun, but we have about 15 minutes left, and I will use that for the exercise 3 to show you a few things. Especially we'll look at an ASEAN phylogenetic tree of this virus. But I just want to quickly recap. So we looked at the concept that you have different regions in a sequence that have different roles, that there are different tools that help you aligning them. And you can, from the alignment, also create a phylogenetic tree that allows you to find some of the relative relations of it. And the important concepts were that you can look at sequence similarity to give you ideas about the ancestry. You need to be careful that there can be a similarity that can occur by chance. And I gave you some hints how to judge when this could happen. And especially for colleagues that are often involved or know already more about protein structure, don't forget you have very valuable information in the sequence itself. You have evolutionary information, you have conservation, you have functional motifs that you can see in the sequence already. So when you're doing 3D structure modeling, docking, etc., don't forget about all the info you can get from the sequences too. And these are the 3 key concepts I want you to take home also today with you. And these tools for sequence analysis can do very relevant work. And I hope that many of you will also sign up for GISET and try out some of the tools that are there. And I will show you some of the things, the trees, as our last exercise on the GISET page. So while I do this, I'll also look again in the chat, and if you have any questions while I'm loading this in the background, please let me know. So for your information, I just went to the gisei.org webpage. If you log in, you can see more things. These are all the tools that you can play with, including a blast search, including some trees, like making the big trees or putting your own sequence into the tree, or looking at the mutations in the structure. You can do all of this here. What I wanted to show you is this part here with the epidemiology. And what I showed to you before was, for example, if you're interested in tracking of different variants, then you see the rise of the Delta variant in the different regions. You can see where they occurred in the map. You can further zoom in. You see now also during the soccer Euro Cup, a lot of people coming together and they're opening up. And but you also see consequently the case numbers rising. And you can get some idea about the percentages of the variants in the different countries from here. You can also, if you're curious and you want to know how good your national surveillance is, you can look at here this genomes in the submission, so like what I showed you before, but it's updated. And so, for example, we can look at the last 30 days. Then we pick ASEAN, and you can have different ways of sorting. So last 30 days, of course, is you need to have cases and sequencing by them. Maybe last 90 days is better to look at. And you see that actually most of the countries are submitting genomes, and the time to deposition is also relatively good. So it doesn't take too long for a sample to be sequenced until it shows up there. This shows you a bit of how the national efforts are contributing to the database. And for the last bit that I'm going through, we're looking now at our ASEAN tree here, because we are all in the region. And the big tree will show the big picture for all the countries in the world. So it will underrepresent the cases we have here. Of course, you see there is also some loading time necessary because the tree itself still has several thousand entries. This tree is made with a maximum likelihood approach. And then the reason why I want to show it is because it has a very nice interactive way of summarizing the data. So I want to ask again, is there any other question for the moment? I will then just walk you a bit through this. So my exercise essentially is that all of you can go there to that site, the phytodynamics. You can also choose the ASEAN tree or any other. And it's just for you to play around a bit. Find the sequences that you have in your country, if you can find some of them. And for example, here in the left panel, you can change color by variant. So we are interested in where are, for example, the delta variants, the alpha variants, etc. And you can see that here colored by the tree. So the delta variant are here, these yellow ones. Singapore had an outbreak with delta variant, and they also did a lot of sequencing. So you'll also find them here. But the one that is highlighted here, for example, is from colleagues from Indonesia as well. And you also see that the alpha variants are here, and the beta variants are here. If we now switch back the color by region, you can see that it takes some time to update. You can see, for example, here, this cluster, this alpha cluster, seems to be particular for Thailand. Then for the delta variants, there are several from Singapore, but there are also several from Indonesia that form distinct subgroups. And you also see that here, this part that was beta, there is this light blue color a lot from the Philippines. And the Philippine genome effort also has highlighted that there are many other strains here. You also have cases from Cambodia here that are also similar to the one, the alpha outbreak in Thailand, for example. And you can click in more detail. What I want to show you is also that by going through this tree, you can also make like a temporal picture. It is not guaranteed that the transmission has actually happened in that direction, because it depends a lot in terms of how intense the sequencing effort is. But I will just play this here for you, and you'll see that it goes through the tree. You see there are different outbreaks, and we are traversing the time here, and sometimes there were connections to the outside, and sometimes there are connections within the region. And these patterns are calculated from the phylogenetic tree, and they help us a little bit in understanding patterns and ways of transmission. And essentially, this helps us that we don't need to make necessarily lockdowns and block everything, all travel, all trade, but rather understand where are the risk factors and then minimize those risk factors so that you can open up in a controlled way. And you see it's taking time to go through the tree. Because there are actually several thousand of them happening at the same time. And I do believe that right now many of you are on the website too. And let me know in the chat if if things do not fully load for you. I see the fact that the animation is slow. It means actually that the load is high, but at least it's still showing. And what you can learn from this is how to control the virus in a way, and how good your measures are in controlling the virus. For example, before the Delta variant came into the different countries, you can check, did I manage to keep it out? It seems this variant is not easy to keep out for anyone. And many of our regional countries already have the most cases, the majority of them are from the Delta variant, because it has some characteristics that make it spread a bit faster. Okay, I see a question. So about the different standard parameter selection, the typical size, so it depends, of course, on the length of the genome that you're comparing and the number of the sequences. For example, MAFT is able to do, let's say, 1,000 genomes of 30,000 on a normal modern computer or even a laptop. But to do this on a larger scale, for phylogenetic, you need to consider two steps, the alignment and then the tree calculation. So about the alignment, when we make alignment of the 2 million entries together, we do this on a 64 core machine with 1 terabyte RAM using MAFT, but not the standard way MAFT is run, but we actually use tricks. We are parallelizing it by making many pairwise MAFT alignments and then stitching it together. The most time it takes for alignment is when the answer is not simple, but when there is a difference. So we first identify the simple answers, and from that build add the more complex ones later. And for the tree methods, for example, the Bayesian approach on a desktop, I would only run 100 to 200 genomes at best. This will still take a day or so. And because you need to run about 10 million trees, 10 million steps in the Monte Carlo. And so for the most accurate method, in the case of genomes, I would say 100 to 200 you can do on a desktop. And if you use the fast methods on a desktop, you can still do 1,000 to 5,000. But if you want to do 500,000, you need to go to HPC in environments. So a second question from Regina, if we also use the sequence masking here. So because the genomes are so similar to each other, there is actually, we are interested in every little difference. So we do not mask out, let's say, low complexity regions, but at the same time, we know that there are some sites that are also the methods for sequencing seem to be calling randomly more often. So we do not use the standard low complexity algorithm, but we use a phylogenetic measure that tells us that this is a very ambiguous position. So it has a very low phylogenetic signal. So we are masking out in our comparison positions that have a low phylogenetic signal. That means they are found in many different parts of the tree. But it's the concept is similar, but because the sequences are very similar, we want to maximize the amount of differences that we can see, even though they may be in a low complexity region. So I think we reached exactly the time. Let me go back to the big view. If you have any questions, so actually I don't know if my email has been posted there. I'll also send you my email here in the chat if you have any questions. So Nisha, I will still look at your question. So most of the that's the answer to Nisha's question. Most of the proteins have a globular part. There is a small part that are non-structured, and but there are most often when the protein has a globular part, it has also a small part that is non-structured. So the BLAST and the sequence similarity method is valid mostly for the globular part and the blossom substitution matrices. However, you can still, if you are careful, can use this information. Like I said, for these viruses, for example, we do not mask out low complexity region, or we are not restricted to just the globular region that makes the protein. We want to get as strong of a phylogenetic signal as possible. So it depends really on more the application that you have and the data situation. So if you have a whole range of very similar viruses, you know they are related. It's not a question, are they related or not, that you need to be careful with the low complexity. It's more like, are the signals that I see true or some technical random artifact? That's the only thing we need to take care of. but because they are so similar, like 98% identical, we know that the low complexity portion is actually not so much of a problem. So I'm aware that you have your lunch break now. I gave you some hints during the presentation that what are typical questions that could be in the quiz. You know, there's just a few little multiple choice questions. Ask the organizers if you don't get the link to the slides. You should have already gotten them. And with those, you can use that as a reference to answer the question. For me, it's more important that you get the concepts and that you see the value in the work. And I was very happy to see all your feedback. So I saved the chat, and I will also pass on extra credits to the ones that had replied fast. I don't know how exactly they're going to use it, but I'm very happy to have had your interaction and your good questions. So thank you very much, everyone, and have a good lunch.
Thank you, sir. So for the students, let's come back online at 11:50 before the session starts. Thank you. Have a good lunch.
Thank you, Sebastian. Good job.
Thanks, Fabrizio. Congratulations to Italy moving on.
Oh yeah, especially with me living in Spain.
Yeah.
Great.
Okay. Thank you. Bye.
Thank you. Take care.